a training where you get everything you want. But I believe this is a foundation for many people that really want to venture into doing molecular biology work, bioinformatics. So just to check, I want to believe we all can hear me.
Yes.
Yes. Are we seeing the slide?
Yes.
Okay.
Yes.
All right. So integration of medicinal chemistry with pharmacology and biomolecular target. It's a well-positioned topic at this point because virtually all we want to learn in terms of hands-on tomorrow have been covered in the first two lectures. But it's very important for us to understand the interconnection medicinal chemistry have with this other aspect of biochemistry, so to speak. And this will definitely help us in not just designing future experiments as far as computational biology is concerned, but also to understand how to optimize the procedures, the processes, and what is best to do at certain point in time. So don't expect any hands-on application much from this aspect. As a matter of fact, this is the point where we actually discuss where bioinformatics actually relies so much on other aspects of biochemistry or molecular biology, so to speak. Why I'm saying this is because in the past, I've actually seen people that just want to learn the skills of bioinformatics for the sake of, now I know how to do bioinformatics, I can design an experiment in bioinformatics, I can get results and then publish it on his own. You would realize that at some point, just gathering results under bioinformatics becomes almost impossible as a standalone thing. There is need to incorporate bioinformatics with other aspects of bioassays. I'm actually talking about not just the in silico now, but the in vitro, in vivo, and the ex vivo aspect. So this time is just for us to discuss how medicinal chemistry brings all of them together. And that is what we want to discuss. That is what this aspect is about. So I'll go to the next slide now. What's wrong with my slide? So what's medicinal chemistry itself? It's just the aspect of chemistry that has to do with designing and developing pharmaceutical drugs. Either you are getting that drugs from natural product or product derived from natural products or from synthetic product. The most important thing is we have a product, be it natural or synthetic, and then we are doing some form of identification, synthesis, and development to bring up a novel chemical entirely that is suitable for specific therapeutic use. And medicinal chemistry sometimes does not all revolve around just coming up with novel drug. There are times that we, in medicinal chemistry, they deal with even existing drugs, explore their biological properties, do some quantity structure activity relationship, and then come up with what we call hybrid strategy to come up with new drug, a novel drug from an existing drug that has already been certified to be potent. So this playing around is what medicinal chemistry is all about. Of course, we can go on and discuss, but that's basically what medicinal chemistry is about. So I'm going to start very quick with interacting with people. And I want us to tell me or show me at this point, if you have anything based on what I ask, you can either type it. I don't know whether Dr. Fatoki or Dr. Anna is around. You can help me come up with that pool. I don't know that pool. So the question is, in your own opinion, or based on your knowledge in the past, how long do you think it takes for researchers to come up with a new drug? So it's a question, it's an interaction. If you want to type in the comment section, you can type in the chat section. If you want to say something, you can raise your hand. I'll call you to talk. If you want to share a link, you are free. So we are interacting on that for 3 minutes. Are we with me, please? Is everyone with me?
Yeah. Yes.
Okay, so opinion, what you know, someone might say it takes 100 years to come up with a new drug. Fine. Okay, someone is saying 10 to 15 years. Okay. Someone is saying 15 to 20 years. Yes. Do we have more opinion? Maybe someone is saying that, okay, 10 years. Okay, 5 to 10 years, yeah. 4 to 5 years. 7 years. Yeah, so it depends on the drug. People are saying sometimes 15 to 10 years till patency is gotten. Yeah. 9 to 16 years. Okay. Do we have any contrary opinion? Thank you everyone for, okay, no contrary opinion. That's good. Yeah. So just before we move forward, recently we all experienced this COVID-19 pandemic, and people talk about, oh, for vaccine to come out, they have to do this and do that. It takes 10 years, this and these years for us to come up with vaccine. But we all know that with COVID-19, I think in less than a year, they came up with vaccines. And some of us saying, oh, actually, they are bridging protocol for you to do this, you have to do in vivo, in this and that and that. But actually, that was made possible with the help of medicinal chemists fully maximizing bioinformatics. So with natural products, either synthetic products or combinatorial chemical libraries, the use of molecular modeling and pharmaceutical biotechnology, there is a way medicinal chemists have come up with a kind of a scheme, a loop, whereby in less than 10 years, a drug can get to the level of clinical trials. And this is the flow whereby there is a screening of a large library for them to get hit improvement, lead improvement, and then come up with a credible drug candidate. Now, when we talk about the hit, it's actually talking about new molecules that have potential pharmaceutical interest. So anywhere you see me mention the word hit, or you see me mention the word hit, I'm actually talking about molecules that are potential drugs. So you see a lot of people using hits, and stuff like that. So there is a place of coming up with it and improvement on it. And from there, the hit molecule get improved, and we talk about a lead compound. Now, for lead compound, we are talking about a hit compound that have been pharmacologically, pharmacodynamically, and pharmacokinetically investigated. And they've realized that these compounds, be it chemical or functional group modification has allowed them to be able to fit suitably for that therapeutic purpose. They must have passed through, the different stage of pharmacology, so to speak. And then from there, we talk about this lead compound leading to being a drug candidate. Of course, that is when they begin to investigate the mechanism of action and then focus more on the target discovery. And then from there, we can talk about preclinical trial and clinical trial. So in a way, when we talk about the integration of, molecular pharmacology and biomolecular target in medicinal chemistry, we are actually talking about subterms of medicinal chemistry. Because what medicinal chemistry talks about basically is understanding molecular pharmacology, getting biomolecular targets, and coming up with something therapeutic. So with this loop of series of experiments and assays, including bioinformatics as the basis, it has actually helped researchers in recent years to come up with novel drugs, novel compounds in few years. In my lab here in the UK, my supervisor and his team actually came up with a compound recently that have this neuroprotective property, and the whole thing, the whole compound was actually concluded and patented in less than 5 years, and it even is part of what I am using presently for my PhD research. So these are examples of the natural product or natural product derivatives that have been established in the past, and we realize that most of them are predominantly, let's say, discovered in the early 2000s, because that was when the field of medicinal chemistry was actually coming to limelight gradually. So this is basically the philosophy of medicinal chemistry, and you can see that the aspect of bioinformatics, heat expression toolkit, working with this chemical library and the likes, is something you can't rule out from medicinal chemistry. We can see that under analysis, we have one of the major things that many people in Nigeria today doing bioinformatics actually apply, that is the QSA, that's quantitative structure analysis comparison. I did something like this during my master's, but it's under the biochemical analysis. So we can see that with issues and hypotheses revolving around this, medicinal chemistry have been able to get to a point where they maximize in silico tools to come up with drug optimization in a very minimal time. In less than 5 years, with the application of this loop, or more of like a dogma, it doesn't take 10, 15 years to get to a point where people establish a drug. And if you look at this point, there is the use of computer-aided drug discovery, and that is what most of us do at the moment in bioinformatics. Either some people use this orthodox Fina, which we definitely install today. Some use Maestro Schrodinger, which I am aware that it's not free, so it's not something that is common in Nigeria. You have to buy with money. So this loop, as you can see, makes up more like what we call the medicinal chemistry philosophy, or the central dogma of medicinal chemistry. And as much as I have said that we need the in vivo and the in vitro assay, the fact remains that bioinformatics play an integral role in the success of medicinal chemistry as a field. So there are subsets of medicinal chemistry. The first one I want to talk about is the computational chemistry. And of course, you must have I've mentioned some terms like each drug, lead drugs and the like. So this is more like the cycle of computational chemistry. So computational chemistry simply talks about using, computers like, aided drug discovery to come up with drug target discovery, to come up with optimization for selectivity, to come up with understanding the molecular basis and even the molecular target that are actually related to certain molecular compounds. And like I've said, it can either be with known structure of target, it can either be with known active molecular compound as your target. It can be a derivative of a known compound. It can even be a derivative of a toxic compound. It can be anything. The idea, the good thing about computational chemistry is, as far as it's computerized, it is very hard for you to go wrong. And what you see, as you can see the flow, it is from this point of knowing your structure of target that then we talk about molecular structuring, or we'll talk about molecular modeling, either for similar molecules or for pharmacophores that are different. But of course, we are talking about a computation at 3D level, where you can see the structure of the compound, where you can see exactly where they bind. The beauty of it is that you see where they bind, you see where they can inhibit themselves, as like it's right there in front of you. So it makes the work proceeding to pharmacology very easy for the researchers that are involved. And that's why you realize that it's easier to proceed in terms of, actualizing or confirming whether a molecule is a potential drug or not. So I want us to discuss a bit. At this point, I want, if you can, Google on your phone or your laptop, and I want us to look at the people that won 2022 Nobel Prize in Chemistry and tell me one or two things you can say about them. So, like I said, this aspect is the only aspect we are not repeating tomorrow, because the hands-on practice on this one is linked to every other aspect we are going to be dealing with tomorrow. So if this is our own hands-on practice, I think it's really worth it. So you can just go to your system, your phone, whatever, and then search through people, the Nobel Prize in Chemistry for 2022, and we just want to discuss one or two things about it. So I'm giving us 3 minutes for that, and then we'll discuss about it. So I'm going to open the floor to people to talk about that. So you can use your phone, you can use your laptop, you are free to use anything. So if there is something you want to say, you can raise your hand. You're allowed. So like I said, what we are briefly looking at are the subset of medicinal chemistry, more like the sub-discourse under medicinal chemistry. And the first one we have talked about is the computational chemistry. And then the second one we want to talk about is what is actually leading to the question I'm asking. So if you have something about it and you want to discuss, you can raise your hand, then I'll ask the admin to unmute you, then you can talk, or you can unmute yourself. So I want us to understand that this is a workshop, and my own philosophy about teaching is I'm not an island of knowledge. I believe I can get something from you that I don't even know before. So this is an opportunity for you to teach me, please. Yeah, so if you have anything you want to say about it, or you saw anything about it, you want to raise your hand, and then I'll give you the floor to talk. Yes? Did anybody get anything about it?
Yes.
Okay.
It's good afternoon. So it says here that it was won by Barry Sharpless, Martin Medan, and Caroline Bertozzi. And says for Caroline Bertozzi, it was because of our work in click chemistry, which molecular building blocks snap together and efficiently. So it says that our work enabled the use of click chemistry in living organisms on the Nobel website.
So what these people did in theory actually looks very simple. As a matter of fact, in theory, it is very, very simple. And that is one of the beauty of medicinal chemistry. So what they did was they have sub-targets. They have sub-targets, and they use different techniques to join these sub-targets together. And as they click the target together, the new compound that is formed was able to elicit a therapeutic effect. I mean, forget about all the big English, but that is what they do. That is what they do. So, understanding the biochemistry of compounds, you can use different methods like the fluorescence, the imaging, the conjugation. So what people do in clicking is more like using medicinal chemistry to come up with synergistic effect. And that is what these guys did in 2022. Of course, they must have started before that gave them the Nobel Prize in chemistry. And this click chemistry we are talking about is one of the aspects of medicinal chemistry. Understanding individual potential of a reagent and clicking them together in the laboratory, either by synthesis or whatever, to come up with what? A new therapeutic effect. And that's one of those things that the medicinal chemists do. And as you can see here, you can use spectrophotometry, you can use MRI, you can use flowmetry, whatever it is, just to bring about the conjugation for the purpose of drug discovery. So another aspect of, or another sub of medicinal chemistry is the application of chemical database. And of course, this one have been, should I say, have been overemphasized today already. And by tomorrow, even from this evening, you'll probably have hands-on practice on it. Bioinformatics has made the research of drug discovery so easy that millions, like for example, look at PubMed. There are 96 million compounds already in PubMed that have been characterized. All you just need to put is the name. It gives you many information about that compound, and it's easier to proceed with your experiment. As a matter of fact, the understanding of the chemical information of a particular compound makes it easier to study either the biochemical properties or the biopharmacokinetics activity, or probably study their molecular targets. So chemical database is one tool that is also commonly used in bioinformatics. As a matter of fact, for every of your bioinformatics work, you would need to always visit the medicinal library. And it's as simple as that. So that's all about that. And then there is another one we call the combinatorial chemistry. So how does combinatorial chemistry come in line with medicinal chemistry? So this aspect of medicinal chemistry make use of large number of molecules, or let me use the word plenty number of molecules. But the idea in this field is not to use all those molecules. The idea is to come up with a scaffolding hybrid, whereby they can screen large library to a little library and then come up with some form of hybridization. Now, this might seems opaque, but I have an example at the end of the discussion to explain that to you. So they usually use this not just for drug discovery, but for modification of lead compounds. You know, I said when we're talking about drug discovery, we talk about identifying lead compounds and then identifying lead compound, and then you go on to preclinical or clinical studies. There are times that medicinal chemists use combinatorial chemistry to come back to the level 2 by playing around the lead modification to come up with another novel compound from the existing novel compound that has improved therapeutic activities. And that's the playing around they do about it. I'm going to show you an example. As a matter of fact, the compound I mentioned that was discovered in our lab here actually used somewhat this combinatorial chemistry. So it's as simple as getting the compound, having a linker, getting a substrate from the linking, synthesizing, breaking up the linker, coming up with a new product. And this is one of the papers I actually tried to look at, and this paper actually explains how they use chimeric compounds to, apply this combinatorial chemistry. And here they use E3 ligand. E3 ligand is one of the very common protein that is known to elicit ubiquitination. There are some kinase dependent pathways that, in a way, after phosphorylation inhibit reaction that may cause that may lead to maybe some form of disease pathogenesis or whatever. This E3 ligand is one of the compounds that actually inhibit that. It's one of the things that I'm actually planning to use in recent time, because I know I'm planning to order some of these things this week. So what they did here is to do some combinatorial chemistry on this E3, link it to a small molecule, do some kind of linker, break it again, come up with something new, and subject it to other in vitro and in vivo assay. So you can go through that paper if you want to read more about that. So that's basically that about this medicinal chemistry. And there is no way you talk about drug design medicinal chemistry that you don't talk about quantitative structure activity relationship. So so this is actually basic. If you have a compound, you want to compare with another compound. You have a compound, you want to compare with a target. You have a target, you want to compare with another target. You know what we mean? Either drug-drug interaction, drug-target interaction, drug-toxic compound interaction, and the like. So statistical modeling using QSAR actually make this thing to be very, very easy. And it plays an important role in the optimization of leading compound in combinatorial chemistry. So this is more like the flow of the combinatorial chemistry. You have therapeutic target, you have a lead discovery, then you have a lead optimization, there is development of new candidate, and then you talk about a drug. And of course, this lead discovery that we have here is more like going back to a loop of the scheme that I showed earlier. So now that is actually, let me say, that is actually all about the sub that we may likely encounter in medicinal chemistry. But then you ask yourself, I know the other two speakers have actually mentioned about medicinal chemistry, having a compound of 10,000 compounds, screening it down, and then you are getting 1, getting 5, working with them, which is basically what bioinformaticians do. But it is not something that they do at random. There is let me just say there is how do I put it now? There is a particular pedestal that needs to be followed. It's more like a landmark that needs to be followed, and that's what we call the high throughput screening. I know many of us must have been hearing about the high throughput screening, high throughput screening. So what this high throughput screening actually does is what we do on a daily basis in bioinformatics: screen larger libraries, use efficient techniques to simplify what you want, and then focus more on the simplified version, and then go on to the discovery process. So this is more like the main thing that has actually made drug discovery so easy for the medicinal chemistry. So there are different methods that are under this high throughput screening. So when you hear high throughput screening, it is not an assay. It is more like a pedestal where you have different assays that can capture under it. And what this has helped us to do is that, unlike going straight to in vitro experiment or in vivo experiment, where you have to do a lot of trial and error, where you have to try a lot of things for you to come to a conclusion, with this high throughput screening, you can do many things at a time, and then it saves your time as well. So you can imagine investigating hundreds of thousands of compounds in a day. This is possible. The only challenge we have is that this equipment can be very, very, very expensive. So recent time now, throughput screening has massively been employed in computer-aided design, in in silico library, and molecular docking softwares have been used together to, in a way, upscale the use of cell-based platform. So instead of us wasting resources, wasting time and everything, just going to, ex vivo in vitro, we use these aforementioned tools to do a kind of a high throughput screening that give us some level of confidence on the efficiency of what we are bringing forward for either predictability or acceptability in the clinical phase. So this is more like the flow chart of what happens. It takes years to get to maybe approval of administration in charge of drugs and maybe post-marketing surveillance. But with bioinformatics being at the tail beginning here, it's easy for us to use bioinformatics tools to get target identification, molecular dockings, to ascertain their target, their interaction, and probably establish their ADME properties. And from there, we talk about in vitro studies. And then there's assay development, which then brings the drug to where we have the high throughput screening. Now, this high throughput screening, there are some sort of in vitro and in vivo assays. And as well, there are molecular modeling that also works here. So this is the point where you have a proper screening of the library and know whether there is drug likeness properties or not. If it's not, you go back to screening. If it is, then it comes to the lead optimization. So it has a rapid migration from being a hit molecule to a lead molecule with the advent of high throughput screening. So the question I want now, these are the classifications. So basically, you can have the in vivo assay or the in vitro assay. It can be cell-based, it can be biochemical assay, it can be on whole organism. There are even some ex vivos that are actually applied in high throughput assays. And for the biochemical assay, it can be monitoring enzyme activity, looking at receptor target binding, or looking at, hormonal regulation. So here we talk about either phosphorylation process modulating something, or ubiquitination process degrading or not changing the conformity of a particular protein, stuff like that. So the question is, I want to ask, and at this point I want to pause and entertain question. And at the same time I want to ask, does anyone here know specific example of high throughput screening methods? Either you've come across it before in a paper, or you have been involved in a research team where they have done it before, or you have a proposal or a research plan where you're planning to do stuff like this. So I want us to interact at this point. And please, I don't know, for few people that know me, I really like it when I interact with people. I always feel sick when I'm the only one talking. So I want people to talk, please. So does anyone here know of any specific high throughput screening method? Any answer that is, oh, this is specifically high throughput assays? Yes? If anybody wants to say anything, you can raise your hand.
Maybe this can come close to it. I'm aware that in some phytochemical analysis, there are dozens and maybe hundreds of compounds that are screened from extracts or plant materials using special techniques that, at times we call it fingerprinting. But the one we normally do around here, maybe people have 10 compounds, 20 compounds, but there are places where they have a lot of compounds from the extract. So maybe that is a kind of a high throughput screening or whatever. It's not to maybe look at the structure, look at any detail, something about the compounds, but to just get as many of them as possible that are in that particular, what do you call it, extract or natural product, whatever. So I think that is a form of high throughput, because I think it is all about getting massive amount of data or amount of particular compound in an entity. That is what all this high throughput something is all about. And I'm aware that in some fields too, like in proteomics, we can look at different parts of the brain, either in a disease state or maybe in a normal state. What are the features of the proteins that you have in the cerebrum, that you have in the striatum, that you have in the hippocampus, in a particular brain that have been subjected to cerebral ischemia? And they can subject it to proteomics and look at all the types of protein, getting large amount of data. So this is what I think I understand by this high throughput, whatever.
Yes, you're right, sir. But the issue with the conventional method that we apply for high throughput screening is that they take a lot of time, a lot of resources, and they require a lot of manpower to achieve them. I'm sure few people actually know that this is our mass spectrophotometry. I know we do that in Nigeria. MSLC, we do it in Nigeria. It's actually an old method of high throughput screening. I was expecting someone to mention that example, actually. It is an example of high throughput screening. It's just that now, because of this more sophisticated methods that are coming up, attention is being deviated away from mass spectrophotometry in most of these advanced countries. It's usually a big, large machine. You have to put a lot of things in the machine and stuff like that. So it's also a method of high throughput screening assay. Thank you for that, sir. Is there anyone that want to say anything else?
Yes, sir.
Okay, sir.
Yes, I think microarray is also one of the examples. Just that one is related to what Dr. Saliu did the other time, that if you have a compound, they will try to inject it across the cell and see which part of the cell respond to that particular ligand. Then they can now focus on that particular area that responds to that particular language. Thank you, sir.
Yeah, that's a very good example. I think that is still very much in play. That is still very much in play. Thank you for that. Yeah, anybody want to say something else?
Please, I have a question, sir.
Okay, sir.
Yes, please. I want to ask, first of all, if there are any disadvantages to the newer methods to the newer output screening, throughput screening methods. So, and the second question I want to ask is that are these screening methods, are they complete substitutes for clinical trials, for conventional that you mentioned in the start, that they take time, the reasons we need faster methods of drug discovery? Are they complete substitutes, or do they just incorporate them into the conventional methods of clinical trials? Does my question make sense, sir?
Yes, it does make sense. So the first one you asked was that what are their disadvantages, right?
Yes, you are right, sir. Yes.
One of their disadvantages is that some of these high throughput assay, the machine that we need to do them are very expensive. Very, very expensive. There is a Ferrastar machine in my lab that I'm using presently. I think the lab bought it at 500,000 pounds. £500,000. You know how much that is in Naira? It's a lot of money. It's more than the budget of some university in 5 years in Nigeria. So that is one of the advantages I know. But the good thing about this thing is that most of their parts have been computerized. So they are easy to use. You see, these days when you go to nature, I mean, some of us have been privileged to see this thing. That's why we know. When you go to nature, you go to all these high impact factor journals, and then you see a lot of results. Trust me, many of them have not done something much. It might just be get one sample, put it in a machine, and that machine generates like 50 different results. And they were like, wow, these people have worked. You know what I mean? Because most of the steps of this machine have been computerized, it is because of that, the human error is minimal. Do you get? You can imagine if I put you in the lab now and I ask you to begin to prepare 10,000 tubes. Trust me, before you get to 500, you will start mispipetting. So in terms of the price, I know that's an advantage. However, every of these techniques are open to improvement. And I know that every now and then there are improvements, there are upgrades. There are even sometimes that the manufacturer will come to the university that they have sold this machine to, and they will tell you, oh, we've just done something in this machine. Let us help you upgrade it, this and this and that. So nothing is actually absolute. For the fact that there is still an input of human, there is still an aspect of human input, definitely nothing is perfect. I don't know whether I answered that your question.
You did, sir. You did. Thank you.
So the second question, if you follow through the flow chart I've actually showed you, I mentioned something at the beginning that I've met people. In fact, I have friends that in the past few years, because I followed some of my friends' publication. I have some of my friends that in the past few years, all they have published is just in silico work, molecular docking. That's all. And I said, in the real sense of it, you cannot get to a point where you begin to talk about either preclinical trial or clinical trial if you don't go to this in vivo and in vitro experiment. So the answer to that question, like I've mentioned earlier, if you follow through very well, is that bioinformatics as an aspect does not replace clinical trial. It doesn't. The only thing they do is that what you need to do before proceeding for clinical trial will be minimal. But there will always be a point where you have to go back, where you will definitely do clinical trial. So I'm trying to go back on this screen. I don't know how to do it. Don't mind me. I'm trying to, okay, so I want to show you the slide I'm talking about. Look at this. Can you see this slide?
Yes, I can, sir. Thank you.
So what the bioinformatics and all those things does is that they make us do less at the in vitro study.
Okay.
So that by the time, whatever we are able to come up with at the in vitro level, we just be subjected to the high throughput assays. And then you can see, if it has drug likeness property, it goes to what? Optimization. Remember, I mentioned eat and live. And if it passes through optimization, it goes to what? Clinical trial. If it is success, they submit it for approval. If it fail, what happens? It go back to what? Library screening, and the loop continue from here. But the good thing is that the loop doesn't have to start from the beginning again. It can just start from this point where you have just to go back and what? Screen the library. And part of the thing you can do in library screening, because these are things that we do. I know I was privileged to work with some bioinformaticians before I left Nigeria. And what you do, instead of going back to the drawing boards, come back to library screening. Since it is FDA or NAFDAC or whatever that disapprove it, what are their approved drugs? You can screen that lead compound with their approved drug. Are there chemicals that are analog of that your lead compound, or that have similar therapeutic traits? You can screen them with those compounds. Are there bioactive compounds or naturally derived products that mimic or that look similar to the drug that has failed the clinical trial? You can screen them with those bioactive compounds. And once you do that, you just come to what? This, from the screening, you come back to the HTS. And you can see that the arrow between the virtual screening and the HTS is reversible. So with high-thorough screening, you don't have to go back to the beginning. It's just a matter of going back by a step or two. So with this, you save years as a medicinal chemist, as a drug developer. That is what we are saying. It in no way, it cannot in any way replace clinical trial. Do we get that?
Yes, thank you, sir. Thank you.
Okay.
Sir.
Dr. Sally, you want to say something?
Yeah, I have a question. It can
Okay.
go to probably Mrs. Ajayi or Dr. Patuki can help me, and also your contribution to. So there is what I know, you know, we have what we call drug repurposing. That is, you repurpose drug to not to follow the what is supposed to address, but repurposing it to another kind of probably ailment or disease. So in the process of repurposing drug, the active ingredient, can we, to start the process of repurposing this drug, does this screening also very much, can it shorten the time to probably get to know if this drug is particularly good for another aspect of disease? I don't know if you get what I mean.
I get what you mean.
Yeah, so can we use drug screening, in silico studies to check if this, to shorten the time to get this drug to be able to probably go to process of repurposing? Thank you.
Yeah, well, I believe it is very possible, because I think the only thing that takes a drug from this point, remember, that is why I initiate us with those terms. Before we can talk about high throughput screening, we are talking about a hit molecule, not just any other molecule. We are talking about a molecule that has already shown some traces of therapeutic advantage. Are you getting me, sir? So it is not a total failure of a drug on its own. And of course, drug repurposing is one thing they do. So it's more like, oh, this drug is doing this, but we want it to do exactly this. Then they do a kind of repurposing. So drug repurposing, they can do combinatorial chemistry with it. They can do click chemistry. You remember the click chemistry I showed you, sir? Okay, probably maybe you want that drug to do 5 things, and the drug was able to do 4 things. For example, you can look for a compound that can do the 5th thing that that drug cannot do, and then you just click them together and then take it through the screening again. Are you getting me, sir?
Yes.
So that is on its own some form of repurposing, just to make sure that the molecule that has gotten to a point of being referred to as a lead molecule satisfy the therapeutic purpose with which we are trying to, address it. So yeah, it's possible for you to have drug repurposing without going back to the beginning. And it's actually it's what we all do anyway. It's what we all do. Even in the basic biochemical analysis that we do, there are times that we do a lot of optimization. There are times that we play around with concentration, with dose, with condition with which we expose our animals. There are times that we play with even in vitro experiment to see exactly what we want. So it's all repurposing. The main purpose is, but then if it keeps failing, then there might be need to take 2 steps backward rather than the 1 step backward that was suggested in the schematic view.
Well done, sir, Dr. Josiah. Yeah, for the drug repurposing that Dr. Salio was talk about, maybe for maybe a existing FDA approved drug that has been used for cancer. There's a case that, okay, after a lot of advancements in diabetes study, now they can now see that, okay, this particular drug that has been known as an anti-cancer agent can also work very well as an anti-diabetes drug. So the same company or another company that discovered that, we try to repackage it and try to label it for anti-diabetic purpose. So that is kind of repurposing. And sometimes the actual approved drug could be optimized, maybe, okay, to improve maybe the efficacy, add 1 functional group, as you have said, in order to improve the efficacy to meet the new indication that it is just being discovered. So I think that is about repurposing, and you are right, sir.
Yeah, and I think that's basically what medicinal chemists do in what we call hybrid scaffolding. I have an example that I will show afterward. We call it hybrid scaffolding. The drug I'm currently using for my research, what they did was actually hybrid scaffolding. It took them a very less time, and it actually won them a patent. They have a US patent for that drug in my lab here. It's something very simple. They just did scaffolding, that's all. Simple chemistry work. I'll show you. So let us go forward. So these are some examples of HTS method. We've mentioned some that we know, including MSLC, fluorescent-based experiment, like using fluorescent dyes. They are the most common one, actually. Superchlomylon dyes, or some radioactive label experiment, like this rubidium and thallium octic acid. These are used to measure they are used to detect the activity of kinases that play significant role in some signaling pathway of human kinase genomes. And then there is another method, there is another technique we call it Aurora ion channel reader. This machine is very, very expensive, but it can be used to replace this radioactive base acid, but it's a very expensive one. And of course, coming back to our in silico modeling techniques, they can also be used to assess the pharmaceutical and pharmacokinetic properties of a lead or hit compound or other therapeutic compound, be it the one we are trying to repurpose or the one we are trying to come up with something new. So these are some other examples. Of course, have I seen all this thing before? No, I've not seen number 2 before. I've not used it before, but the number 1, I've used almost, I've done like 4 or 5 of that number 1 in my lab here. So I know. And the funny thing is that these are experiments you can finish in less than 2 hours. But they are very, very expensive. That's it. They're very, very expensive. So these are examples of high throughput assay readout. I think here, I suspect that they use fluorescent and they also use, microarrays here. And just from one readout, they're able to get different results. So one of the disadvantages as mentioned here is that it doesn't give good highlight of complexity, but there is high throughput, there is high yield. So I don't want to waste our time, but here I actually wanted to ask, do we know anyone or has anyone been involved actively in high throughput screening experiment before? Or maybe I should ask, do we have anyone that has been involved in any high throughput experiment apart from our facilitators? Do we have anyone? Yeah, so I'll just go ahead anyway. So here, it's just talking about, there are a lot of human genome. What led to that drug I'm about to explain is there are a lot of human genome, they want to screen it, they want to bring it down to, understanding how this kinase signaling pathway works. And then we know basically that for proteins, their functions are easily modified by kinases. So when there is phosphorylation of protein at certain sites, their activity change, their enzyme activity, their cellular position, their association with their other protein, their conformation, even their 3D structure change. I'm sure that all of us, when we're doing, how do I call it? This central dogma, DNA to RNA, RNA to protein. There is an important aspect we always forget, post-translational modification. And that is actually the point where most, how do I put it, most physiological defect occur. I know there was a time, sir?
I said, yes, you are correct, sir.
Yeah, I know there was a time, I mean Professor Kimarado and Dr. Sally will bear witness. We submitted a paper, and then I think they're asking us that we should go and do Western blotting to know the protein level. They say, they are not even willing to collect or to accept the how do I put it, this RNA expression from us. Because a lot of things happen during post-translational modification. And that's the idea behind this point. So in our lab, we are more looking at what happened after phosphorylation or ubiquitination. So there is a target at this protein called SPARCO-SR1. This is ST20 related proline and alanine rich kinase, or this one we call it oxidative stress response 1 kinase. I don't want to bore us with this, but what happened is, in a layman language, when you have this chloride transporter in the body and you are well, the moment they are phosphorylated, you become unwell. That is just the simple English. So they've been so keen at targeting this wing-spark pathway, and how their phosphorylation affects physiological activity and causing different pathology in brain, heart, kidney, and the likes. So that was actually what led to the development of this ZT1A compound. So originally, for all these kinase, for all these kinase activator, so to speak, or this wing-spark signaling pathway, when they phosphorylate this ion channel, they cause pathology. And there have been many drugs that have been developed in the past. One of the review I wrote when I newly came here, I explained like maybe 20 or 50 of those compounds there. I could share it maybe later. So what they did here, like what Dr. Fatoku was saying, what they did here was that they look at some of the inhibitor of this kinase that works very well, and they selected these 3. I'm telling you the aspect of the medicinal chemistry in this work now, and they selected 3. So one of the challenges that they know that this drug, truly they can inhibit this pack. They can inhibit SPAC. But then they discovered that none of them is ATP. Is none of them, all of them, they keep binding to the ATP. And when they bind to the ATP, the energy with which they used to act will reduce, because instead of them to focus on the phosphorylation, they will keep battling with ATP. So what those medicinal chemistry do was to help us come up with a compound that will be non-ATP competitive. And that's how they came up with this Z1A. So simply what they did was to do some chemical reaction, some synthesis in the lab to come up with this compound. So that's the compound. The reason for this A was that when they were doing the experiment, they came up with like A, B, C, D, E, F, G, H. They came up with like 8 of them from different hybrid design of these 3 compounds, which are already kinase inhibitor, but they are ATP competitive. So that is simply what actually what medicinal chemistry do. They look for something that has been working before, sometimes do some hybrid design, come up with some scaffolding to come up with another one that is a bit better than that. And these are actually shown, it has actually shown wonderful results, because they went ahead to do other experiments, which we can refer to as throughput experiment. Protein expression was investigated. Then they look at the IC50 and SPAC kinase. This was where they did all these rubidium uptake, thallium uptake, and then kinase activity as using the Ferrastar machine that we have in the lab. And then they did fluorescent polarization too, and some pharmacodynamic study, and some in vivo and ex vivo experiments. So these are some of the results that was actually obtained there. I think this is the fluorescent polarization that was done. They saw a lot of similarity for the cache type that is actually responsible for the activation of those kinase, Then subject this Z1A to a lot of screening after inhibition. And look at it. They subjected the compound to many things. And they realized that it doesn't bind to anything apart from the Wnt kinase. Whereas other potential drugs that were developed before them binds to every other thing, even those ones that they were not supposed to bind to. So what makes this thing novel and special is that its specificity for not binding with ATP. So sometimes medicinal chemists can go extra mile just because they want to achieve one single thing, which most times I will tell you is worth it. And these are some of the results that was obtained from it. This paper was published in Nature Journal. It's rather unfortunate that I was supposed to join in this work, but COVID delayed me in Nigeria back for a year, and I missed out of it. It's something that's always painful, but it's all right. So basically, that is actually what happened in medicinal chemistry. So I want to stop here and entertain question. One thing we should understand, I want us to take home is medicinal chemistry will always try to come up with drug candidates, go back to do the development of lead molecule, do thorough post-screening before clinical studies and the likes. And then the interaction of this field with chemistry, biochemistry, pharmacology, and medicine itself contribute to successful drug design. And there is no how we talk about effective drug designing without good knowledge of bioinformatics. So it's actually a highly prized aspect of science at the moment, and we all should just sit back and watch the space at which bioinformatics is going. because it's actually really making research easier and quicker to attain meaningful results. So this is where I'll stop at this point. These are some of the resources I'm happy to share anyway. And that's it. If you have any question, the floor is open now. Thank you. Dr. Ojo, I'm done.
Thank you, sir.
You're welcome, sir. Is Prof there?
Yeah, I'm around. Thank you.
Yeah, I'm done, sir.
Thank you very much. Okay. Thank you. Do we have questions? You can unmute yourself and ask, or you can drop your questions in the chat box. Anyway, I think we can move to the last speaker for today.
Sir, the click chemistry was the question later addressed. You asked that question.
I asked it. No, it was not addressed.
Okay.
So click chemistry, is it using our mouse to just click? Pang, 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 pang?
No. It's actually it can either be in the wet or the dry lab. It can be in the wet or the dry lab. It's just about understanding the, how do I put it now? The orientation of the compound in relationship to the understanding of their pre-existing therapeutic potential and linking it with another compound. It's more like a linker, more like a synergistic new product that can enhance the therapeutic potential of the new product from the two forming. And somebody was saying something about this bioorthogonal chemistry. Is actually what one of the things I actually read about, but because of time, we couldn't just go through everything. So basically, what medicinal chemists were doing is just to look for compounds, see how they work, how they can improve them, and the likes. Like, okay, I just want to go there. So here, sir, like what we are saying is this is the work that won Nobel laureate in 2022. Can you see my slide, sir? Can you see my slide, sir? Hello, sir.
Thank you very much, Dr. Joseph.
You were saying we are going to say thank you.
Okay, yeah. Maybe I should just stop here.
Please, Dr.
Please, it's like somebody wants to ask some question.
Yes, sir, please.
All right, go ahead, please.
Please, earlier in the lecture, you talked about the COVID vaccine. I am aware that I know that clinical trials, like you said, that they are not HTR is not a substitute. I know that clinical trials can still take so many years, between 10 and 15 years on like in many cases. How were they now able to still get the COVID vaccine out in such a short amount of time? If this was not, since they still have to do, I assume that they still have to do clinical trials.
Yeah.
So how were they able to get it out in time? So
one of the techniques they used actually, because that time a lot of drug actually came out. One of the techniques they used was what Dr. Salio actually explained to us. Some of them actually employed the technique of single-strand RNA to arrive at something quick.
Yeah, so can I?
Yeah. You can come up, sir.
So yeah, like Dr. Desire said rightly. So like, you know, when the COVID-19 SARS-CoV-2 came, it's a novel virus. So nobody knows anything about the virus. So they have to know what the virus is, what are the genetic components and everything. So to know this, they have to sequence the RNA, the RNA of the virus, to really understand what are the genes involved, what does it mean, what are the cells, what are the genetic components of the cell of the virus. So they have to sequence it, understand the genes, understand genetic composition before they can say, let us apply this kind of thing to see if probably it will be able to address this genetic alteration in genetic profile and all that. Yeah.
So it wasn't a trial and error, whatever. It was actually a throughput experiment that was done. Okay. It's just that in recent years there are techniques that makes this output faster. It doesn't make them a fluke or a fake. So I don't know other people, but I know, I think, is it not AstraZeneca? I know they really explore the techniques of that ssRNA to come up with something meaningful in a short time.
Yes, sir. Also, these days we have what we call in silico clinical trial. So this pharmaceutical company, they have data of different genetic factors of different set of population. They have them already in the database. So what they normally do is that they will do what we call in silico clinical trial to see the result. So once they get the result, now they can, this set of population, this is what we should expect. This set of population is what we should expect. So they do that kind of in silico clinical trial in their pipeline to see, where and what side effect they should expect. So by the time they go to feed, they already have a kind of a expectation. Yes. That, this is what we are expected to get. If they don't get it, then they can now easily deviate and do other things. Thank you, sir.
Thank you very much.
And I know that time they did test those vaccine on some people. It's just that the normal clinical trial where they say, try it for a year, try it for 6 months, they didn't go through that, but they actually tested them on human being before rolling them out. Because for the fact that it passed the in silico clinical trial does not mean they should just go ahead and release. So I know they tested it on some human sample, but not for a very long time because of the urgency, because of the fact that they needed the drug in circulation urgently.
Thank you very much, sir. Thank you. I understand. Thank you.
Dr. Abuki.